Okay, there's a lot of information on this slide regarding MHC molecules, but let's talk a little bit about those and their importance for the immune system. MHC molecules are considered self-antigens, and they actually have the ability to display other proteins on their surface. So it's like a double protein antigen structure. Okay, MHC proteins kind of have a groove in them and on their structure, and their groove can hold either a self-antigen or a foreign antigen. So remember, MHC proteins are glycoproteins that stick out of the surface of our cells, and they either display self-antigens to let the immune system know everything is okay with the cell. So it's a way to say, I'm okay, this is a normal antigen, everything's good. Okay? Or they can display non-self or foreign antigens, which let the immune system know that there are foreign invaders in the body. And there are two classes of MHC proteins. There's MHC class 1, which is this type of protein is produced by all nucleated cells in the body. Then MHC class 2 proteins are produced by cells that are called antigen-presenting cells. Antigen-presenting cells purposely eat foreign invaders by phagocytosis to let immune cells know of their presence in the body. The APCs we have in the body, believe it or not, B cells fall into that category, but macrophages also do this, and dendritic cells, which are macrophages that are found in a lot of mucous membranes and areas close to the surface of the body. Okay. MHC molecules are genetically determined. So we have a huge amount of variation in the MHC molecules in the human population. It's incredibly rare for two people to have exactly the same MHC molecules, except if you have an identical twin or triplet, okay? People that are genetically similar, so if they are related, relatives are more likely to have similar MHC molecules and therefore be a better match for tissue transplants. I bring this up because, in part of the normal process for immune surveillance of the body, the lymphocytes will go around the body, particularly the T lymphocytes, and check MHC molecules to see if they belong in the body or not. So if an individual receives a transplant from another individual and they have not checked the MHC molecules, then there is opportunity then for the T cell to recognize that the MHC molecule is foreign and attack that cell. So that is what T cells and immune system cells are checking to see if a cell belongs to the body or not, is simply by that MHC molecule. Now, if an MHC molecule is on the surface of the cell and has a self-antigen displayed in its groove, then the immune system will leave that cell alone. But if a cell has an MHC molecule displaying a foreign antigen in its groove, then that is a signal that that cell is infected and we need to remove that cell. So let's take a look and see what I'm referring to here. So it says foreign proteins or self-proteins are presented at the cell surface by MHC class 1 molecules. So here is a cell of the body, and it could represent any nucleated cell in the body. And so it says here is a foreign protein or a self-protein that's inside the cell. It doesn't matter which one it is. If it is foreign, this can happen. If it is a self-protein, it can happen. You'll notice that the cell will chop up the protein into fragments to produce antigens. They get transported to the rough endoplasmic reticulum of the cell. From there, the antigens are going to combine with MHC class 1 molecules. See that little groove I'm talking about in these molecules? Okay, so we create this complex where there's an MHC class 1 molecule and a protein in the surface. That's that double protein thing I'm talking about. Okay, then if you follow number 4, it says the MHC class 1 antigen complex is transported to the Golgi apparatus, packaged in a vesicle, and then transported to the plasma membrane. Okay, so here is where we have two scenarios. In the case that protein is a foreign antigen, So let's say this cell was, infected by a virus or something, and this is a foreign protein, then this combination of a self-MHC class 1 molecule but a foreign antigen means to the immune system that this is an infected cell and we need to destroy this cell. In the other scenario, if this is a self-antigen, a normal protein that the cell typically makes, and it is displayed on the surface of this cell by a self-MHC class 1 molecule, then that says it normally does not stimulate cell destruction. So the body knows that there is no need to destroy this cell. It's healthy. No need for destruction here. Okay? Now, just in the last slide, I mentioned that if there are foreign MHC molecules, then potentially a T cell could come to this cell and look at the MHC class 1 molecule, and regardless if this is a foreign protein or a self protein by that cell, then that could stimulate cell destruction. All right, let's see what's going on with the MHC class 2 molecules, what that's all about. So we said MHC class 2 molecules are produced by antigen-presenting cells. And so in the case of an antigen-presenting cell, which again is more or less going to be a macrophage, or a dendritic cell, which is a macrophage that's typically found in the skin and some mucous membranes, okay? The macrophage will purposefully eat a foreign antigen by phagocytosis, bring it in via a vesicle, chop up that foreign antigen into little parts, and this cell can create MHC class 2 molecules, combine the vesicles, and you'll see that the foreign antigen will be attached to the MHC class 2 molecule. Again, this has a groove, that purple thing has a groove in it, so it can hold foreign antigens. We will then transport the MHC class 2 molecule to the surface of the cell, and we have the foreign antigen sticking out of this. This will stimulate immune cells. The whole point of this, okay, is to let the immune system know that there is something funky in the body and we need to search for it. So the end result of this antigen presentation is not to kill this antigen presenting cell, but to alert the immune system that there is something wrong in the body. There's something foreign in the body. Okay. Now let's keep going here and see what we do a little bit more specifically. Okay. I will do this in the next video. We'll start at co-stimulation.